This is Chapter 4 lesson where we continue to study the organelles found in different types of cells. We're going to start looking at vacuoles. There are different types of vacuoles, and in a previous lesson we did mention the food vacuole, which is derived from part of that organism's membrane of the cell and utilized to contain a particle of food that is then further broken down by lysosomes, that cleanup crew, and then any waste materials are exported from the cell, but some of those food particles can remain within the cell to provide a source of potential energy for the organism. So if you're not familiar with that food vacuole, we'll make sure you go back to the lesson on the endomembrane system and the nucleus that we discussed that a little bit more in detail and had some diagrams to go along with that. But there are other types of vacuoles, and like the food vacuole, these are also membrane-bound organelles. They're going to be found in eukaryotic cells, not prokaryotic cells. And we're going to look at two different examples, the central vacuole and the contractile vacuole. The central vacuole is going to be found in many plant cells. This organelle is mainly storing water in addition to other nutrients and minerals that might be in that water, such as organic compounds. It's there because it might be holding it onto it for later purposes as a storage vessel, but also when these central vacuoles are filled with water, they kind of push out on all the surrounding parts of the cell, the cytosol that's around it and all the organelles inside, and that puts ultimately pressure on the cell wall that's just outside of the plasma membrane. So it causes this phenomenon that's called turgor pressure, and plants actually like to have that turgor pressure. To be turgid means that it's rigid and firm and it can stand up tall against gravity to be able to get light for photosynthesis. And we'll study that concept more, that turgor pressure, in a later chapter, chapter 5, when we look at how materials can enter and exit the cell. Specifically, we'll look at how different solutions of different salinities will influence how much water is either absorbed or lost from a cell, such as a plant cell. And we'll even look at that in lab. So you wouldn't have any central vacuoles in animal cells, only in the plant cell. Another type of vacuole is the contractile vacuole, and this is not found in most cells, to be honest. It's only in very specific types of organisms, such as paramecium, that live in a freshwater environment. So their bodies are constantly absorbing fresh water from their surroundings. The contractile vacuole acts like a pump to actively pump excess water out of the paramecium so its body doesn't explode because of too much water rushing in. Now, this process requires energy on behalf of the paramecium. So in order to access that energy from the food that it eats, it needs another organelle to do that. That brings us to the mitochondria and also the chloroplasts, which we'll talk about after mitochondria. The mitochondria are going to be the sites of cellular respiration, which is the metabolic process that uses oxygen to generate ATP. That ATP is then utilized by the cells, such as the paramecium, to be able to operate that contractile vacuole to actively pump water out of its body. We'll study the mitochondria in greater detail in a later chapter. All eukaryotic cells are going to have mitochondria. So I'm going to skip ahead to show you a depiction of a eukaryotic animal cell first. See if you can find the mitochondrion. We've got one here. And you're looking at like a cut through of it, so you can see the infoldings, and those are the surfaces where the ATP is synthesized. There's another one over here. Okay, so that's an animal cell representation showing the presence of mitochondrion. And similarly, for the plant cell, we also have mitochondrion. But there's some differences here between the animal and the plant cell that we now need to mention, and that in the plant is the addition of the chloroplasts. You're not going to have chloroplasts in the animal. Now, there could be other eukaryotic cells with chloroplasts, like macroalgae, for example. Chloroplasts are going to allow those organisms that contain them to be able to make their own food through the process of photosynthesis. That food can then be used as a source of potential energy for the mitochondria going through cellular respiration. For a plant, they can make their own food and then harness the energy in that food. For an animal who can't make their own food, they have to consume that food somehow into their body and then harness the energy through the cellular respiration process that happens in the mitochondria. I just want to bust the misconception right now that like chloroplasts would only be found in plants and mitochondria only in animals. It's not true. Mitochondria could be in all eukaryotic cells that require the production of ATP. Some cells are going to have more mitochondria than others depending on what their function is. But if they need to be able to make ATP, they need mitochondria for a eukaryotic cell. We'll look at some additional differences between plant and animal cells in our next little mini lesson.